This is John Pan. I'm the founding medical director for the newly established Office of Integrative Medicine and Health at the George Washington University Medical Center. I have the privilege of bringing you this webinar, The State of Research in Homeopathic Medicine and Understanding the Biological Basis for Homeopathic Remedy Responses. This is sponsored by the American Institute of Homeopathy. You will hear from two of the world's most renowned researchers and practitioners of homeopathic medicine, Doctors Iris Bell and Dr. Peter Fisher. This will review the research findings in the field of homeopathic medicine and our developing understanding of the biological basis for homeopathic remedy responses. This will be a 90-minute session. Each speaker is allotted 35 minutes. And at the end, we will have 15 minutes of question and answer. Dr. Fisher will be our first speaker, followed by Dr. Bell. Dr. Fisher is the Director of Research and Consultant Physician at the Royal London Hospital of Integrative Medicine, the RLHIM, in London, England. He is board certified in homeopathy and rheumatology. The RLHIM is part of University College London Hospitals, one of the largest academic medical centers in the UK, and is Europe's largest public sector center for integrative medicine. He also serves as physician to Her Majesty the Queen. Additionally, he is member of the World Health Organization's Expert Advisory Panel on Traditional and Complementary Medicine, and helped to draft its traditional and complementary medicine strategy for years 2014 to 2023. Dr. Fisher.
Good morning. Thank you. It's a pleasure and an honor to be here in cyberspace. Yes, thank you. Incidentally, the World Health Organization has just changed their traditional and complementary medicine department to traditional, complementary, and integrative medicine in the last month or so. So that's a step in the right direction. So what you can see on screen here is our hospital in London. We're right in the center of medical London. Just here on this side is the National Hospital for Neurology and Neurosurgery, one of the leading UK centers for neurology and neurosurgery. And right behind us is the Great Ormond Street Children's Hospital, again, the leading national center and a leading international center for pediatrics. So, and this is Queen Square. We're very central in London, maybe 300 yards from the British Museum. Although being stuck between two larger and richer neighbours is not always a comfortable place to be. So I'm going to try and give you an overview of what's happening in research in homeopathy. A lot going on, so I'll try to keep it concise. So the first thing is to be clear what homeopathy is. It's much misunderstood. Indeed, a few years ago, we did a review of the safety of homeopathy. And actually, the most amazing finding was that actually about 40% of the products where safety concerns had been reported and which were alleged to be homeopathic were not. They were herbal. And really, this is by expert people who really should know better. So homeopathy is much misunderstood. And that's perhaps because it's an idea rather than a technique, an idea. It's the idea of treating like with like rather than herbal medicine, for instance, which is the use of plants or plant extracts. It's not. It's an idea. It's the idea of treating like with like. This idea was propounded by a man called Samuel Hahnemann at the very end of the 18th century. Sometimes, well, quoted in Latin, similia similibus currenta, which means let like be cured by like. So that is the basic issue, a basic idea of homeopathy, and it's in the name. Homoios as in homogeneous or homosexual, meaning same or similar. Pathos as in pathology, meaning disease or suffering. So same disease, same suffering. So that is the basic idea of homeopathy, and one should never forget it. The controversial part of homeopathy, the reason that it attracts so much scientific skepticism, is its use of very small doses, including so-called ultramolecular dilutions, where, theoretically, no molecule of the starting substance is present. So this is clearly a problem, but actually it's a problem that, that we're making great progress in solving. It seems that it isn't a fundamental stumbling block, despite what certain skeptics would tell you. And Iris will be talking a bit more about that later. There are other ideas, including holism. The idea of constitution in homeopathy, we sometimes say, we don't treat diseases, we treat people. So, we can't focus, for instance, one of the areas where homeopathy has a lot of potential is AMR, antimicrobial resistance. And there, we are not focusing on killing bacteria or viruses, we are focusing on strengthening host immunity, strengthening resilience. So you're treating a person, not a disease, not a bacterium, not a pathophysiological person, a whole person. And that second word, terrain, that is not terrain, that is terrain. Terrain, a French word, meaning quite literally soil. It's the idea of seed and soil, where the soil is the person, is the patient, the seed is the disease, certain seeds flourish in certain soils. So a very similar idea. And then the sensitivity is very important in homeopathy. In some of the biological systems we'll look at, you find quite consistently that if it's a healthy, balanced system, nothing happens. The system has to be, in a sensitive state, knocked off balance, sick, otherwise it won't react. It will not work on a healthy system, on a healthy person. If you give the wrong homeopathic medicine, nothing at all happens on the whole. You have to have sensitivity. So that leads to the idea of idiosyncrasy. It has similarities with so-called type 2 adverse drug reactions, the idiosyncratic as opposed to the dose-related drug reaction. Individualization is very important in homeopathy, and again, sensitization if we're talking about biological experiments. You must sensitize your system or it won't work. So there is a related concept, is hormesis. Hormesis is simply the stimulatory or beneficial effect of a low dose of a toxin, as opposed to the linear threshold or non-threshold model. So what you get is a J-shaped or hockey-shaped curve here, for instance, as opposed to the linear threshold or non-threshold model would predict a straight line from here to here. Whoops, wasn't meant to happen just yet. So a straight line from here to here. Or if it's a threshold from here to here. But actually, this is now an old slide, this is an old, but it shows the principle nicely. It is very, very widely observed that paradoxical reactions happen at low doses. And since a toxin is by definition bad for you, then a paradoxical reaction is salutary. So this, just to give you one example, this is the effect of low doses of the insecticide chlordane on the rate of weight gain of crickets. Crickets are insects. Chlordane is an insecticide, therefore you would expect it to kill them, which indeed it does at doses above 100 parts per million. But at 1 part per million, it is doubling their rate of weight gain. You can observe similar things with physical agencies. This is the effect of low-dose gamma radiation on the rate of weight gain of guinea pigs. And this is very, very widely observed. If you want to read it up, Calabresi, the name to look for is Edward Calabresi, professor at the University of Massachusetts at Amherst. I don't know if you've seen the bibliography that Iris has distributed, but just look up Calabresi. He is the Pope of hormesis. And this has been observed in tens of thousands of cases because you can see it. For instance, industry has to do large-scale toxicology screening for substances they want to use. So there's huge databases, and if you analyze it, very, very, very frequently you find this paradoxical reaction at low doses known as hormesis. Oops, wait a minute, sorry about that. Pressed the wrong button. I have to go back, sorry. Sorry about that, let's go right back. Sorry, I pressed the end instead of the next. getting the free preview. Okay. So this is the controversial part of homeopathy. The preparation, the very high dilution. So this is how you prepare what we call a 6C. C stands for 100. So a 6C dilution. You start off with a mother tincture, which is defined by the pharmacopoeia. There is a US homeopathic pharmacopoeia, exactly what it must be. You mix it with 99 parts of water and ethanol and shake it vigorously, so called succussion. For insoluble substances, you use a process called trituration, which is grinding. And I think Iris is going to talk a bit more about that later on. But this is what you do with soluble substances. You shake it and repeat that process 6 times for a 6C. Now, we know that Avogadro's number, or to be precise, Avogadro's law and Loschmidt's constant. Avogadro was an Italian who invented the principle. Loschmidt was a German or a Czech or an Austrian, depending on your perspective, who actually discovered the number. The important thing to remember is it's of the order of 10 to the 23. That means that if you were to repeat this twice for a 12C or 10 to the minus 24 dilution, it is extremely unlikely that particles could persist, although there actually do seem to be exceptions to that. In some cases, particles do persist for reasons we're starting to understand way beyond the Avogadro limit. But this is, of course, the controversial part. It means that in these high dilutions, which are used quite regularly in homeopathy, there's very unlikely to be any molecule of the starting substance. So I'm going to start from a very pragmatic point of view, sociological point of view. We'll move from the sociological through the clinical evidence and then to some of the biological model evidence. Who uses homeopathy? And what about the results of family physicians who incorporate homeopathy in their practice to compare to those who don't? So the recently published systematic review of the use of homeopathy, 11 countries, there's good data from 11 or 12 countries. In fact, France isn't listed there actually, but there is good data. It was excluded from the study for purely technical reasons. The data is good quality. So overall, worldwide, 1.5% of people consulted a practitioner in the last 12 months, 4% purchased homeopathy. Much higher, for instance, in France, 10% of the population and nearly 20% of children per year are consulting a homeopathic physician. Also very high in India. We don't have reliable figures for India, but it's certainly extremely high. There are 250,000 practitioners. I don't mean patients, I mean practitioners of homeopathy in India, and over 350 homeopathic medical colleges. So it's extremely widespread. We don't have good data. In the States, there is very good data analyzed by a prestigious center. This is done by Michel Dosset and colleagues from Harvard, based on the National Health Interview. So that's the federal interview that is done every 5 years. So good quality data, good team that analyzed it. Just over 2% of the US population used homeopathy. This is in 2012, 15% increase since the previous one. And a pattern that you observe that has been observed everywhere, or at least everywhere it's been looked for, in Germany, in France, and now in the United States. Users are female, healthier lifestyle, and have relatively high education. The same, very similar patterns have been found in the past in France and Germany, and more recently in the States. The most common uses for upper respiratory tract infections, and homeopathy users often use multiple complementary therapy, but they like homeopathy particularly when it's prescribed by a practitioner. So this is to show you, yeah. So this is just my favorite little thing about education. Users of homeopathy in the States and elsewhere tend to have higher education, above college education, essentially. You can see the data there. And this is the perceived use, this is the reported use in the National Health Survey. Here you have homeopathy prescribed by a practitioner. Here, this is homeopathy self-prescribed. This is supplements. This is osteopathy and chiropractic. So health conditions a great deal is this one, important in maintaining health and well-being. So as you can see, compared to other
complementary therapies, it's perceived by the users as very helpful, particularly when there's a prescribed as opposed to bought themselves over the counter. Just to look at cohorts, this is the comparative effectiveness study of homeopathy. Comparative effectiveness studies are non-randomized. It means that you take cohorts of people who attend, doctors who incorporate homeopathy in their practice versus those who don't. And it's a large-scale study, over 6,000 patients, done by the University of Bordeaux in France. And it compares us conventional mixed practice and homeopathic physicians. And these were the cohorts: musculoskeletal disease, upper respiratory tract infections, sleep disorders, anxiety and depression. In terms of clinical benefit, medical care and medication, adverse effects, loss of therapeutic opportunity, and economics. Loss of therapeutic opportunity means, were the people with anxiety and depression going to commit suicide while they were waiting for so-called effective treatment? The answer is no. Actually, ironically, some years ago, we did a study of homeopathy head-to-head with Prozac, and we had a terrible time with the ethical review board, or I think you call it institutional review board in the States, who said, oh, well, what happens if they commit suicide in the meantime? As we now know, they're more likely to commit suicide with the Prozac than with the homeopathy, but anyway. So this is just to show you the diagnoses, so over 6,000 patients, and patients tend not to consult. For cardiovascular diseases, they tend to go to conventional physicians. For anxiety and depression, for musculoskeletal disease, and for upper respiratory tract infections, they are more likely to go to see homeopaths. So there is a difference in practice. And of course, that is the weakness of clinical effectiveness studies. The two groups don't match. The strength is that they compare what goes on in the real world. So just again, the demographics, again, my favorite thing, higher education, healthier lifestyle, belief in holistic and natural treatment, and a positive attitude to participating in their own care. There's a question there, which direction does the causality go there? Is it because the homeopathic physicians give them advice on participating in their own care, or were they more inclined to do it anyway? The truth is we don't know the answer to that, and it needs further investigation. But actually, in terms of their comorbidities, the main things, the main pathological parameters were very similar between groups. So, the results are comparable. There we are, just to show you. So healthier lifestyle, less likely to be overweight, less likely to smoke, more likely to have a college education. So this is musculoskeletal disease. It's the only cohort I could, there's several cohorts. I'll just talk about this one. Patients did not differ, except for the chronicity. The homeopathic patients tend to have more chronic conditions. Similar progression. Functional scores were very similar at 12 months, but a huge difference in the use of non-steroidals, and indeed in ordinary analgesics, but particularly in non-steroidals. This is just to show you. So this is the acute situation. As you can see, the homeopaths are using just over half the number of non-steroidals of the conventional doctors, and the difference is the same. It is almost exactly, precisely double the number of non-steroidals used by conventional physicians. And non-steroidals, as you know, are dangerous drugs. They can cause hematemesis, they can cause renal failure. They're pretty pernicious drugs, actually. So that is a worthwhile benefit. Another study, I don't think I've got it here. Oh, yes, I have. Yes, sorry. Also in the same series, showed a huge difference in the number of antibiotics, 43%. They were using less than half the number of antibiotics. It's possible that there were a small excess, that some of the patients did need antibiotics, a small number. It is possible. But an enormous difference in the use of antibiotics. And given we have a crisis in the world with antimicrobial resistance, that's very significant. And some of them, maybe 10% and the 5%, 10% needed antibiotics. Fine, but not 100%. And then the cost effectiveness, basically the homeopathic physicians spend more time with their patients, which costs more, but prescribe many less drugs. And on balance, there is a 20% savings. It's highly significantly cheaper, the homeopathic. So the homeopathic physicians were getting the same results much more cheaply, and many less dangerous either on the individual level, as in the case of non-steroidals, or on the society epidemiological level, as in the case of antibiotics, many less drugs. And this cost doesn't take into account treatment of adverse drug effects. It's just the direct costs. How much did it cost? The consultations cost? How much did the prescription cost? And net is a big saving. This is in the French health system. So that's the conclusions. Less expensive, and this is of important interest. A rather similar study done in Germany, funded by the Krankenkasse, the health industry. Here you can see it, typical, and this is the bread and butter of primary care, I think you would agree. For children, allergy, atopy, basically. This is the typical things I think that GPs, that family physicians see every day. And they looked at the quality of life, severity of symptoms, and costs. And actually, there's an interesting story behind this. This was actually funded by the insurance industry, and they had a hidden agenda, which was they know that patients like using homeopathy, and therefore they would like to offer it in their insurance packages. The question is, would they have to charge more? That's what they really want. That's what they were really interested in. And the answer is they don't. It doesn't cost any more, but gets better results. Just to show you, let me just show you the graphic, So this is the SF36, this is the physical component, 6 and 12 months. As you can see here, you have a ceiling effect. They could hardly get any better. They were pretty much back to normal, much different from the conventional treatment. The mental component, the homeopathic group started off worse and ended up better, although it's not statistically significant. The first 6 months here, are highly significant. And this was corrected for educational level, because they had the same thing. They were more highly educated and tended to be female, but particularly they were more highly educated. So that was corrected for. So that's the conclusion. Basically, better outcome compared to conventional medicine at similar total cost. This is a study, a real-world study. So this is a purely uncontrolled study done at a good centre, the University of Bern in Switzerland, looking at, basically they had 230 consecutive kids with acute otitis media, and they gave them homeopathic treatment. If they weren't pain-free in 6 hours, they gave them the second homeopathic treatment. If they weren't pain-free at 12 hours, they got an antibiotic. So in fact, 72% had resolved within 12 hours. The remainder got antibiotics. And although this was not controlled, this resolution is considerably faster, about 2.5 times faster than what you would expect. And again, there were cost savings. And on the basis of this and other work, the Swiss federal government commissioned a health technology assessment looking at whether they should incorporate homeopathy and several other forms of complementary therapy in their compulsory insurance. This was where they looked at upper respiratory tract infections and allergies. As you can see, fairly even distribution between typical upper respiratory tract things. I don't think there's anything very surprising there. So where compared to conventional treatment, 6 out of the 7 studies showed equivalence. 1 did not. And this was a brave study, I think actually an unethical study. I don't think it's ethical to withhold penicillin from patients with streptococcal infections. So I think that's a bit dubious myself. Of the 16 placebo-controlled studies, 8 favoured homeopathy, 4 showed a trend in favour of homeopathy, 4 showed no advantage. So 24 out of 29 trials were positive, and they said, so their conclusion was probable effectiveness. And that might sound a bit weak, but actually they only had 3 grades they could use: probable, possible, unlikely. So it is the most positive of the 3 grades they could use. And on the basis of this, the Swiss government has now incorporated homeopathy along with acupuncture and osteopathy and a couple of other, Ayurveda, I believe, in their compulsory national health scheme, insurance. Okay, so now as we're moving sort of slowly towards the more basic science, so let's have a look at randomized controlled trials, systematic reviews, and meta-analysis of randomized controlled trials. So you will frequently hear it said, there is no evidence for homeopathy. Well, you can go to this website yourself. You're welcome to use this if you want. This is a live link. This is set up by the Carstens from the Carstens Foundation in Stuttgart, Germany. Karl Carstens was the president of Germany in the 1980s. Serious guy. And he endowed this foundation when he died. And you can access it, no charge to access it. You have to register, but you don't have to pay. So, you know, if you don't believe me, go and look at it. It's probably gone up. In fact, I believe it's now 11:39, so I haven't looked in the last month. Of which 500, nearly 500 were randomized controlled trials, mostly against placebo, some against standard treatment. One of the interesting areas is there's been a couple of studies of Prozac, homeopathy versus Prozac for depression, both of which have said it's at least as effective. Overall, 41% are positive, 54% were inconclusive, usually because they were underpowered, and 5% were negative. And actually, if you compare that, you can quite easily look up, you know, what is the overall conclusion of systematic reviews of randomized controlled trials of conventional treatments, and it's actually remarkably similar. It is rather, you know, very similar. So this is, you know, in terms of how many are positive and how many are not, it's very similar to the overall picture in conventional medicine. So this is a fairly recently published study by my colleague Robert Matthey. This is actually an open access journal. You can read this for yourself. You can get it online. Looking at the best quality studies of randomized controlled trials of homeopathy. They found a total of 32 randomized controlled trials in 24 conditions. And what is interesting is, so this is the odds ratio. This means there is no difference between placebo and homeopathy. Anything on the right means homeopathy is better. Anything on the left means placebo is better. As you can see, there's nothing on the left. And this is the mean value. And rather unusually, it moves away from the center. Usually increased quality is associated with less positive results. Not so in homeopathy. And I have to tell you, in this area, two of the three studies were by Iris. So congratulations, Iris, for your high quality studies. As I say, you can access that online. You can read that for yourself. So just to show better, so with increasing quality, the odds ratio, the 95% confidence will get wider because you've got smaller numbers, but the mean actually moves to the right rather than to the left, which is unusual. So just looking at comprehensive systematic reviews of homeopathy, there's been quite a lot of them. So this is homeopathy as a whole for all conditions. So all homeopathy. So here we have, so this is clearly positive, tentatively positive, inconclusive, tentatively negative, and negative. There's a story to be told about that, but I better not get into that now. And this is homeopathy as a whole. This is just individualized homeopathy. And this is the one we just looked at just now. For groups of conditions, a lot of work. The biggest single area is allergy. And here we have 3 studies in allergy, all of which are tentatively positive. Arnica, we'll look at that. Arnica is controversial because you have arnica negative here, inconclusive here, but this is the most recent one, and we'll look at it in a moment, and it is tentatively positive, so there's some disagreement. This is old, this is 1998, and this is the most recent one published, I believe, in 2015. Again, more, so diarrhea, the work of Jennifer Jacobs at the University of Washington, Seattle, vertigo, rhinitis. This is David Taylor Riley's work on rhinitis. Ileus, fibromyalgia, oscillococcygeal, a series of, you can see it, well, you can see it yourself. This was fun. I was a co-author of this study. It was a Cochrane review, but a quick one. He did it for his PhD, Rob McCarney. And actually, there are no studies. The reason it's inconclusive, there are no studies of homeopathy for dementia. Now Cochrane won't let you do that. But at that time, they would let you do an empty review, which is a quick way of. Anyway, so that's just to give you the overall picture. We can provide references for all of those. So this is one of the ones that was clearly positive. This is the work of David Riley in Glasgow, Scotland. And it's a simplified model of homeopathy. This is isopathy, meaning not like with like, but same with same, treating the same with the same. So what they did was quite simply, they got actually the skin testing stuff. They used to test for skin in the UK, so a typical mixture of grass pollens from a typical UK summer, and they potentized it, they diluted it. And so they did hay fever, and they did one for asthma and one for perennial rhinitis. In this case, they individualized the allergen. As you would expect, it was mostly house dust mite. That is what you would expect from the literature. It's the most common allergen. And statistically highly significant. When you pool it all, I believe it's a total of 250 patients. Highly significant. I can show you the graph. So here we are. So this is the subjective, the visual analog score. So this is hay fever, this is hay fever, this is asthma, this is perennial rhinitis, and this is the composite. As you can see, in all cases, the homeopathy is more positive. And this is the pooled analysis, clearly positive. They used objective outcome measures, so the number of escape antihistamines, bronchial responsiveness to histamine, and nasal inspiratory peak flow rate. These couldn't be pooled because they're dissimilar. You'd be trying to pool apples and pears. But as you can see, again, in every case, it was favourable to homeopathy. So let's turn to, but how am I doing for time, by the way? Let me have a look. Yeah, okay, I better get a move on. So again, no evidence. This is again the Carsten-Stiftung different database. This is called Hombrex. The other one was called Corhom, but it also maintained by the Stiftung, the Carsten-Stiftung. Over 2,000 biological models, 55% of them. Actually, this is slightly out of date. These were done on the same database but a few years ago, so the distribution may have changed. But, whoops, sorry. Okay, yeah. So that's that. Sorry, I've got to get a move on. This is the work. This is the hallmark, of course, of good science is replicated experiments. So this guy, Christian Endler, published in a book now 21 years ago, well, 23 years ago now, but this was the 21st anniversary. It came of age, so it was coming of age. They reviewed what had changed in those 21 years. They found in 2016 126 replicated studies, and 70% of them were reproducible. The null hypothesis, in other words, that high repetition would not show a difference, was refuted by 70% of the experiments. This is Jacques Benveniste. It's the only thing most people know about homeopathy. Jacques Benveniste, who died in 2004, distinguished French immunologist, who did this work on the human basophil degranulation test. Let me just show that to you graphically. So this is a basophil. On its surface, it has IgE, which recognizes specific allergens. In its cytoplasm, it has granules which contain histamine and other immune mediators. So if you get an antigen, it will bind with the IgE, and so let me do that again. It will bind with the IgE, and that causes the basophil to activate. And when it activates, and the same thing can be done by anti-Ig, which is non-specific, will bind to any kind of Ig on the surface. And when it activates, then the histamine comes out into the supernatant, and you get a feedback inhibition loop. So now the mast cell is deactivated. It's a simple cybernetic feedback loop. It deactivates the basophil. But it turns out that the ghost of histamine in a 16C dilution, does the same thing. And this has now been repeatedly validated. Just to show, I'll skip through that, but just to show you that I don't have much time, but believe me, it's been repeatedly validated. That's, I believe, 13 out of 17 experiments were positive. Another simple one done by Paolo Bellaviti. This is the so-called light-dark test. It's a standard test for anxiolytics based on the fact that mice are timid creatures, and given the choice between staying in the dark and staying in the light, will tend to stay in the dark. Anxiolytics such as buspirone, any anxiolytic, will increase the time it spends in the light and the number of transitions it makes. And that's just a solution. This is gelsemium, widely used homeopathic medicine. In high dilution, has effects similar to buspirone, bigger than buspirone here. This is the time it spent in the lit area. This is the number of times it went in and out. So this is a certainly ultramolecular dilution of gelsemium. It has a much bigger effect than buspirone. And then one of the problems with homeopathy is, in many cases, the basic research doesn't quite meet up in the middle. There's a gap in the middle between basic research and clinical research. But I think we're getting close to bridging that gap in this area. Arnica montana, used for trauma, probably the most prescribed of all homeopathic medicines. This is a study done by Paolo Bellaviti, high-tech PCR quantitation of gene expression. So this is just to show you Arnica montana. So you have two types of macrophages, the normal one, inflammation, the one in wound healing, and it is known, what gets, what changes. This is just a quick schema to show you, what is the difference, what is upregulated, what is downregulated in macrophages differentiated to those two types. This is another one where they did the whole genome, big study. And this is, so 7 genes were upregulated, 13 were downregulated. I can just show you this graphically. This is taken from a much bigger table. They looked at it in, I think, 7 or 8 different dilutions. But these are ultramolecular. This one is certainly ultramolecular. So these are the genes that are downregulated, these are upregulated, and this is statistically significant. And it is remarkable that it's almost the same whether you've got substantial amounts of arnica present or very high dilutions. The pattern is very, very similar when you're using it in a herbal or a homeopathic form. High-tech, big-scale study, and it's in +1, so you can access it. I've got the reference here, I hope. Yeah, there's the reference. And then there's the Yaniti study. We mentioned it briefly earlier. Systematic review saying that arnica is more effective than placebo for several conditions. The problem is the wide variety of preparations, doses, and conditions, which vary with very variable outcomes, which make meta-analysis not possible. But the conclusion is it does seem to work at least for some conditions. Just to show you, this is just they had 28 in all. I just snipped out a few. So they looked at herbal and homeopathic. These are all homeopathic. This is in tonsillectomy. This is musculoskeletal trauma. This is joint knee surgery. Just to give you an idea, but there is actually more work than you'd think. And then finally, just to finish off, I rather like this model. This is rather a sweet model. This is based on treating, you stress, again, very important, you must stress your seeds, or not stress them, the control is not stressed. And then they exude liquid, they collected this liquid and evaporated it, and looked at the pattern of the polycrystalline structures in the residues, and analyzed it by an objective measure by this image analysis software. So just to show you, significant differences, but only with stressed seeds. Let me just show you the picture, it's easiest to see. So this is unstressed, this is stressed, this is the stressed seeds. As you can see, there's a difference here between here and here. This is one, whoops, I lost that down. Yeah, that is the one you need to look at. So this is the stressed seeds treated with the active treatment compared to the unstressed seeds compared to the control. So funny little model, but it seems highly reproducible. And these are very serious guys. This is the group led by Stefan Baumgartner, based in Switzerland. And they work very hard on it. And then just finally, a method that shows in differences between very high dilutions of what we call natrium-muriatic in homeopathy, commonly known as salt, sodium chloride and lithium chloride, where they get these dilutions, they cool them to very, very cold temperatures, 77 degrees Celsius, which is Kelvin, which is the temperature of liquid nitrogen. And they do it in heavy water, dideuterium oxide, because it gives a cleaner signal. And they bombard this frozen water with quite hard radioactivity, gamma rays or X-rays, and that excites the electrons, and they jump from their quantum ground state to a higher state. As it is warmed up, they drop back to their ground state, and as they drop back, they emit light. And so this is just to show you, this is the total light emission, which occurs after you get these things and warm them up from 77 Kelvin to about minus 50 to 200 Kelvin. Big difference. So this is shaken water. They shook it, so that is controlled for that, versus lithium versus sodium chloride versus lithium chloride. As you can see, a big difference in the total light emission as it's warmed up. I can show you that another way. So here we are. So this, you start off at 70 Kelvin, you come up to 210 Kelvin, so that's whatever it is, minus 60 Celsius. And this is, oh no, sorry, sorry. What's going on here? Let me get rid of this. Sorry. So this is what happens as you warm it up. This is lithium, this is sodium chloride, this is plain water. And I'm told by people who are experts in this method that this is recognizable. This is the signature of lithium. You can recognize that as lithium. So that's just a method, and this has been reproduced independently. So just to summarize, I'm running late and I'm going to be in trouble with Dr. Pan. Patients who use homeopathy tend to be highly educated, have a healthy lifestyle. Clinical effectiveness studies show better results. Health technology assessment is positive. Economic analysis is positive in France. Systematic reviews. There are a lot of clinical trials, nearly 500 randomized controlled trials, 44 systematic reviews, some of which are positive, including the high quality randomized controlled trials, and particularly in allergy and upper respiratory tract infections. Animal models. We didn't have time to look at this one. We looked at mouse anxiety. Nerve regeneration is another interesting one. They all require pre-sensitization. And then the fractal dimensions we were just talking about. There's low temperature thermoluminescence. And there's a commercial for you. I'm editor-in-chief of this journal. If you really want to know, here it is. It's published by Elsevier. You'll find it quite easily online. And, oh, look at that. And I thank you for your attention.
Thank you very much, Dr. Fisher. I hope, and indeed for a very illuminating talk. And times is running short. I will introduce Dr. Bell, who is a psychiatrist, a university professor, and has been a researcher in areas related to complementary and alternative medicine for 30 years. Dr. Bell has served on the faculties at Harvard Medical School, University of California, San Francisco, and the University of Arizona. She graduated magna cum laude in biology from Harvard University and then received her PhD in neuro and biobehavioral sciences and MD at the Stanford University. She has published scores of papers and book chapters on her clinical research, in addition to a monograph on environmental illness. She was chosen as one of the best doctors in the Pacific region of the US in 1996 and then in 1998 of the entire US. Dr. Bell.
Whoops. Okay. Hello. Thank you for this opportunity to speak with you. And thank you, Dr. Fisher, for an excellent summary of so many areas of research. What I'm going to do today is to take the discussion to the small level, the smallest, most common level of biology, which is actually the nanoscale, and speak to you about more of the physical chemistry findings and then how that will relate to the actual situation in biology and how the reactions occur. So I won't go into anything about the clinical, but the obvious question that comes up from people who are not familiar with homeopathy, and many who are, is this can't work because there's nothing there. You diluted the higher potencies, the higher homeopathic potencies, so much that there's nothing left. Therefore, this is all placebo. And much of the recent research has gone into trying to understand why the dilution does not remove the information from the remedy, even if it removes what we would call the bulk of the original source. It does leave a trace behind, and I'll show you data to support that statement. And what we're going to talk about, somewhat about the structure of these remedies, the signal properties, and then the nonlinear properties of the body as a complex adaptive system. So there are many references listed here. You're welcome to use the bibliography that I posted to take a look at them at your leisure later on. But basically, there is support for this notion that remedies actually may bring structure to the solutions in which they find themselves. Both water itself and actually any solvent, whether it's ethanol, water, or water alone. They also have been demonstrated to generate electromagnetic and optical signals. You saw some data from Dr. Fisher, excuse me, a minute ago. And these signals, whatever they are and however they are, appear to trigger a self-regulatory adaptive response in a cell or an organism. And then that cell or organism takes the healing process from there as a complex, organized system. And we'll cover that in more depth in a little while. The basic concepts of homeopathic remedy preparation, just to review what Peter talked about, are that you either take insoluble material and grind it up in dry lactose called trituration, or you make an ethanolic tincture of an herbal product or sometimes an animal byproduct, and you prepare that to remove the liquid material from the source. You then put it through a serial dilution and succussion process. And literally, when this was first developed 200 years ago, it was done by banging the container of the liquid against a book or some other fairly hard item in order to produce essentially within the liquid of the vial. Excuse me. Now, what is important in homeopathy in emerging research is that there's a lot of research showing findings at the nanoscale. And I wanted to just quickly put it into perspective. It's something like a virus can serve, it actually fits the size of 10 to 150 nanometers. You can see that a DNA helix is actually 2 nanometers. An atom is 1/10 of a nanometer. So we're down here working below the level, for the most part, than of bacteria or certainly a human hair size. We're below the micro level, we're in the nanoscale. Well, I'm not going to ask you to just believe me. There are now multiple replications of the finding, started first from an Indian research group in 2010, that the source material, how it happens, is broken down into and then transferred into even these higher potencies in very crude nanoscale form. So you're seeing very irregularly shaped nanoparticles at extraordinarily high electron microscope magnification levels that were first documented in 6 different homeopathic remedies at both 30C and 200C potencies, again, far above the Avogadro number level. One of many replications at this point, sorry, I may need to move the picture over there. One of many replications was done with copper as the source material. And what is notable here is that not only do you have nanoparticles, which are traditionally considered materials that are in size below 100 nanometers diameter, but you also have a subset of materials that are found in homeopathic that fall really below the, they fall below 10 nanometers, but even sometimes below 1 nanometer in diameter. These are referred to as quantum dot sized materials because they literally produce quantum mechanical effects at the scale because of their uniquely small size, where the atoms of the source and their associated electron clouds are trapped and generate quantum mechanical phenomena when the electrons move around. We, in our lab, did a replication study as well, looking at silver as the source, which in homeopathy is referred to by Latin argentum metallicum. And we found at all potencies, both the 6C, which is below Avogadro's number of fusion, and the 30 and 200C, we definitely replicated a finding of nano-sized materials, but each potency seemed to have roughly the same size of material, of nanoscale material there. The succussed materials did have some nanoparticles, and even the unsuccussed control had some material in it that was at the nanoscale in size. Obviously, there is a significant difference between the verum remedy particle size and the unsuccussed control. So in answer to the question of, well, this stuff is just diluted, there's nothing there, and it's just water or it's just solvent, it is not just water or solvent. It is different in terms of what material persists. But there's more to it. One of the characteristic things that researchers do in the nanoscience world is that they will characterize the surface properties of the nanoparticle to see how stable it is. This, among other events, includes measuring zeta potential of the particles. And if the value at an absolute value is either greater minus 30 or plus 30 millivolts, you have a more stable particle. And what we found was when succussion had occurred, we had more stable particles than we did in the unsuccussed control. So even though there were particles that had different surface properties if they were succussed, and the most stable material of all was actually the argentum metallicum 6C. So very striking finding. We then replicated this finding with a plant-based material, gelsemium, that I think you just heard about in terms of its effects on rodent anxiety behavior. And again, we found nano-sized particles, that were fairly significant in concentration compared with the succussed controls. We ran an additional unsuccussed control here, which was made with a silicon stopper to the glassware. Now, the classic way of making remedies involves glassware and the use actually of natural cork stoppers. Well, cork is made from the bark of oak tree, and it actually constitutes an herb or a homeopathic remedy that has biological effects. What's very striking here appears to be that the cork, in the classical way of making a remedy, may be a participant in stabilizing the particles that are formed during succussion. And if there's any kind of jostling of the material during the testing or preparation, then you're probably going to get some cork extract into the liquid solvent, which will somewhat confound the picture of the controls, the even unsuccussed controls compared with the verum. When you use silicone stoppers, you still have some particles in there. Again, those could be contaminants from a variety of sources. But look at this very striking finding. The silicone stopper nanoparticles are profoundly unstable compared with anything that was deliberately succussed or the unsuccussed control that had a cork stopper. Now, again, keep in mind that any kind of jostling of the glassware, even in transit to the testing laboratory, could have essentially inadvertently shaken or succussed all of the solutions that were on their way to the testing lab. But even then, at least, the silicone stopper produces a whole different kind of particle with its property compared with others. One of the other things that was hypothesized about the same time by John Ives et al. At the Samueli Institute was that the silica, there might be silica coming off the inside walls of the glassware during succussion. They subsequently demonstrated that this was the case with both borosilicate glass and, I believe, soda lime glass. And this was replicated by several other laboratories, including this one in France, where they looked at the succussed material, just looking with pooled samples at material that was succussed in glassware as opposed to material that was succussed in plastic. And you see, now silicon is the element that is silica. Silica is silicon dioxide, and it is formed on contact with oxygen. So the silicon that might be released from glassware during succussion under ordinary atmospheric conditions is going to form silica. But in any event, you can see that there is a substantial presence of silica-related materials when something is in a classical way. The next question was, so what? Because remedies are believed by homeopaths to have quite different effects, depending, again, on the individual who might require them. So what you see here is a study demonstrating that the silica that may come off the glassware, in this case, natrimur, again, this is sea salt. In this case, this is kalimur, or potassium chloride, prepared homeopathically. So the salt is dissolved in this solvent and shaken, prepared with the cereal dilution and shaking that you expect to see with homeopathic manufacturing. And what this particular group demonstrated as a follow-up to some of their early electron microscope work was that the particles of the source material developed a silica coating around them. Now, in addition to the cork I mentioned a minute ago, silica is demonstrated in the mainstream nanoscience literature, nanotechnology literature, to be a very good stabilizer of other nanoparticles. So here you have, again, another stabilizing influence on the homeopathic source nanoparticles that comes from the actual way these remedies have been classically prepared. Now, does this succussion business matter? We know that we're going to release the actual we're going to release the actual glassware components into the solution. Well, there was a group, again, an in vitro kind of study using cell culture where they created an artificial wound between the cells and had them connect with each other, essentially the healing process with cells moving across the gap of that wound. And they compared the material, which was a homeopathically prepared mixture of plant-derived homeopathic remedies with succussed solvent and unsuccussed solvent. And what they found was that the biggest healing effect you see here with the actual Vera material, but with the succussed control, they saw a small amount of actual healing of this wound area between the cells and the cell culture, and the least effect from unsuccussed solvent. And this is compared with a regular negative control, I believe. So again, it didn't heal all the way back to a fully confluent area of cells, but it did heal and was quite comparable to a positive control. Now, again, one of the arguments is, this is ridiculous. This has to be a contaminant. How could you be getting nanoparticles of source material in there if you're just diluting things? Eventually, you've gotten rid of it, haven't you? And that's an assumption. Well, one group that was one of the first to demonstrate the nanoparticle findings followed up a study where they hypothesized that the actual succussion process produced layers of nanoparticles and which caused material to collect in certain areas of the container. And what they actually demonstrated up to, I believe, at least C potency, again, past the theoretical Avogadro's number for this kind of material, they demonstrated that the nanoparticles were in fact transferred, especially when they were sampled from a layer developed towards the top of the liquid material. So that is one way they can get there. It was demonstrated that this is a method, that even if they go through these 15 separate dilution steps of 1 to 100 dilution every time, or 1 to 100 ratio every time, they still end up with layers of nanoparticles of the source material, which they can then sample and move on to the next and the next. This obviously has implications for manufacturing in that there may be quite a bit of variability depending on how a company makes its remedies. There is also other research in modern nanotechnology demonstrating that when you create any kind of agitation or turbulence in a solution of salt, you create nanoparticles and you simultaneously embed them into glassware. They then subsequently come off the inside of that glassware back into solution over time. So you have not the direct transfer possibility, you also have just a release from the glassware. And in homeopathic manufacturing, when they get into making higher potencies, they switch to a technique where they start reusing the same piece of glassware over and over, simply pouring off the last set of solvent and adding in fresh solvent. That means that what they end up with is quite a bit of material potentially left over in the same glassware as they go to so-called higher potencies. So, as we were discussing even in Peter's talk a moment ago, remedies appear not just to be nanostructures, but they appear to have signal properties. And it turns out that nanostructures can generate electromagnetic signals, and even, as I mentioned earlier, quantum entanglement if they are small enough and below, say, 5 to 10 nanometers in size. This was a study done by a Nobel Prize winner from France, where he studied homeopathically prepared bacteria. He actually studied several different types of bacteria, and he was able to demonstrate, at least in certain dilution homeopathic potencies, if you will, using his particular detection method, that there was a unique signal found, at least between, in this case, would be an X potency or D or decimal potency between an 8 and an 11 D or X. So this was a ratio of 1 to 10 dilution in his preparation method. He went on to demonstrate that these bacteria could actually transmit information about their state to other dormant bacteria that were similar to them. So they couldn't transmit to bacteria that weren't of the same ilk, if you will. There had to be a similarity between the bacteria putting out the signal and the bacteria receiving it. This is again, now we're moving into yet again another study involving optical phenomena. This was more recent work done by a different research group in Germany, where they found what they called delayed luminescence with high potency homeopathic remedies. These were profoundly high potency. They took, now I don't know whether this was made clear, but homeopathic remedies, among other forms, are often made by pouring or spraying or drying the final liquid material on the outside of a globule or pellet that's fairly small. And they compared the control globules, which had no such material on, with Argentum metallicum at very high potencies, and a plant remedy called Cantharis at profoundly high potencies. Two different techniques, one of them being the method where you are reusing the Korsakovian method, which reuses the same type of glassware. And what you see is that there is a unique type of signal that the remedy is exuding or emitting compared with the control. So again, we have evidence of a signal property of even these very high potency materials. There are several different groups that have studied ultraviolet visible spectroscopy absorbance with homeopathic remedies, and this particular one demonstrated for a remedy, I believe that this would have been aconite, 30C potency studied with multiple trials, compared with placebo studied with multiple trials, that there was a significant difference in the absorbance when the material was the verum plant, up to a 30C potency, far past Avogadro's number of 10 to the 23rd. This would be essentially 10 to the 60th, I believe. So how can a living system detect remedy signals? And I don't have very much time, so I'll go through this quickly. You'll have many references to take a look at. There is demonstrated evidence that remedies, when without being put in the nose, can stimulate the response of the brain. We demonstrated this repeatedly in our laboratory. We also have multiple studies suggesting that the cell danger or stress response signal network is activated in the immune system, excuse me, and in biological cell-to-cell signaling. So what you may have here is a cascade of events. The remedy shows up as information to convey to the cells, which then start talking to each other and carry on the information. So it may not be that the remedy is forcing something to happen the way a pharmaceutical drug would. It may simply be it is setting a process in motion. And the cell stress response, when it is too strong, kills the cell, kills the organism. When it is a mild stress, however, it actually induces adaptation. And that's what's particularly important, because these are biological adaptations that take place, and it increases the resistance not only to the exact same material, this isopathic type of stimulus, but also to any kind of stressor that is cross-reactive to it. And think about the fact that when we choose a homeopathic remedy for a patient, we're trying to match the symptom pattern of that individual to the appropriate remedy based on what that material could cause in a healthy person. And we're trying to reverse that in a sick person. Again, I just want to emphasize this point that nanoparticles have been demonstrated to activate this cell danger response network. It starts recruiting the dendritic cells in the immune system and inflammation, and starts sending homeostatic danger signals around the system of the body. Cytokines are released, and so on. This was one of the earliest studies in 2008 demonstrating that the gene expression profiles of, again, macrophages, which seem to be key to how the remedies set things in motion, actually will activate in unique ways a homeopathic remedy with mostly plant material, but this is a very complex mixture of homeopathic remedies is used. Clearly, this material called Canova is used in some countries to treat patients with cancers and HIV/AIDS. Now, this is, you would say, well, this is implausible. You still have a small amount of nanoparticles. Well, there's a recent study in conventional nanoscience where 1/10 of a nanomolar of gold nanoparticles given acutely in cells was shown to have up and down regulating effects on gene products. The chronic use also produced effects, but we're more interested in the acute for its parallel to homeopathy, because when we have a remedy, we're giving a dose in a very discrete manner, and we're mainly managing not total amount of remedy, we're managing frequency of the signaling, because we're spacing out one dose from the next. So this is, in one sense, a summary slide. On the left, you see a diagram of some of the components of the cell danger or stress response network, ranging from reactive oxygen species and heat shock proteins and so on, all the way around the system. We have phagocytes, macrophages, D and T cells, central nervous system involvement. And this model is what is believed to be involved simply in the stress response of cells. Well, in the homeopathic literature, if you dig around, you will find papers demonstrating the activation of reactive oxygen species, modulation of heat shock proteins, macrophage activation, cytokine release, and again, in our study, central nervous system changes measured by EEG activity. Nanoparticles are known to be able to modulate the system in different directions, either towards pro-inflammation or anti-inflammation. So let me just move ahead quickly to, you say, well, look, this is a small signal, so what? Well, there are 3 different demonstrated mechanisms of response application that all of which require that you are dealing with a complex adaptive system. If you give it to a simple system, you won't see this kind of response necessarily. One is stochastic resonance, which is a very peculiar phenomenon, repeatedly demonstrated, again, well outside homeopathy, where if you give a noise, a broad spectrum noise with multiple frequencies in it, it will amplify a signal of something else that may be present as a frequency within the larger band that the noise contains. So you're using noise to amplify. Well, remember again, as Peter mentioned, you have to have a stressed organism in order to have an effect. A stressed organism amounts to the biology of the existing disease. And what you see here is a model, really, for amplifying the remedy signal when it is in sync or a similar signal to one frequency within larger disease noise. it then will be amplified by the body itself, not inherently within the material itself. Now, Peter mentioned hormesis, which is another major phenomenon. It may be an umbrella phenomenon for some of these things I'm discussing. And important point here, you see hormesis below the no observed adverse effect level. So you see it down here, and you see it going in the opposite direction to where the toxicity of a material goes. So homeopaths will frequently use a toxic material, but prepare it homeopathically, and presumably are activating a reverse direction of biological response by virtue of the dose. And nanoparticles can cause hormesis. This has been done in multiple studies. A very recent paper, again by this amazing group in India, has taken a look at the effect of homeopathic nanoparticles and their ability to activate a hormetic type of effect on a cell preparation at all potencies, 6C, 30C, and 200C. In this case, again, using silver nanoparticles, in this case, or tin nanoparticles, and not every material produced this effect, but it was observed. And again, you see an improvement in the activity in a certain concentration, and as you get to higher concentrations, a reduction in the activity. So this is hormesis as anticipated by the theory that homeopathy may mobilize by that method. This is a study actually in bacterial metabolism, where one group pushed a known toxin for these bacteria, formaldehyde, and just kept pushing it through homeopathic dilutions all the way to, I believe it was a 20C. And what you see here is a very interesting sinusoidal dose-response curve, where it is toxic at certain concentrations, it gets less toxic at others, and so on and so forth. And this pattern actually keeps repeating through a series of dilutions. This is implying that homeopaths may need more research on the proper potency to choose, because they want to improve the overall effect on the individual without making them sicker. And again, this becomes, it invokes a clinical phenomenon known as aggravations, where some individuals, when they get the correct remedy, first get worse and then they get better. This would be the initial response of the organism. And what we would be looking for would then be, what does the organism do with this once the initial effect has occurred? We demonstrate a somewhat related phenomenon in brainwave response, that when we had people in a laboratory situation, we demonstrated that the people who got the correct verum remedy, the people receiving active remedy versus placebo, actually showed an increase over a 3-month period when they sniffed the correct remedy for them. The people doing just placebo sniffs actually went down. And this was controlled just for sniffing. So this was not just a sniffing phenomenon. These were fibromyalgia, fibromyalgia patients. We replicated and extended this in people who were chemically intolerant, having them repeatedly sniff their remedy. And we found, again, sort of a sinusoidal dose response by repeated exposures over the course of one laboratory session. And did this with both the remedy sulfur and with the remedy pulsatilla. And we found that the people who were high in chemical intolerance, they're considered highly sensitizable people who will react to many different items. And those people seem to show a very different response from the people, the individuals, who were not particularly sensitive to materials. So again, we have responses in humans showing this. Now, finally, I wanted to make a last point, so I hope we'll have a few minutes for questions. And that is, I keep mentioning that the living system is a complex adaptive system. And what you have to keep in mind is that some of the original thinking, not only in homeopathy, but also in traditional Chinese medicine, has been a vitalistic approach of seeing sort of a top-down organization to the system that you are trying to treat. People in conventional medicine are very mechanistically oriented and tend to see, well, we'll break it down to the smallest parts, understand mechanisms, and then sometime later we'll rebuild it. The reality of physiology that has been demonstrated completely outside the field of homeopathy is that the living system is a complex system. It means that there are interactions between the parts locally. They interact with the emergent global property of this particular organism. Then the global property of the organism also has an influence back down on the parts. So it's a much more interrelated network of connections and effects than you typically see hypothesized in vitalism or in conventional mechanistically oriented studies. And again, parts in a complex system, they have interconnections, interactions, and interrelationships that generate the properties of the global system. You know, we as people have very different properties than our livers or hearts or lungs. And complex systems interact by nonlinear dynamics, where small changes can cause large effects in the system if the system is poised to respond in that manner. For example, if the stressor that arrives is recognized by the system as something that could be dangerous at a toxic level, even if it saw a mild form of it, that's the nature of hormesis, you would see an adaptive cellular response to prepare for the future possibility that there might be a toxin there. And the complex adaptive systems are self-organizing and have no single control point as a general rule, but there are some hubs that are somewhat more active. This is a study actually in a yeast model where they demonstrated that if they gave a large stress, they broke up the network associations and interconnections of the system. But if they give a more moderate stress, they see reorganizations of the linkages between the different components of the system. Presumably, this would be what goes on with healing when someone receives a homeopathic remedy. So the question is there any evidence of this kind of biphasic response in people? Well, we did a study where we gave people a dose of one of two homeopathic plant remedies, either Coffea cruda from coffee or a toxic plant called Nux vomica that has strychnine in it. Prepared at the 30C potency, so it wasn't toxic, it was homeopathic. And we gave it to them at bedtime. They had a history of insomnia when they drank coffee. And we made a specific effect on normal sleep staging. It really was an influence on the ability to sleep compared with the placebo nights in thin subject design. But we also did a complex systems-oriented analysis of what is called multiscale entropy of the brain themselves during sleep. And we found that on the remedy night, each of the remedies pushed the complexity either up or down. But on the next night, when no remedy was given, there was a rebound effect in the complexity of the brainwaves during sleep, slow wave sleep. And so you see here, very complex study, very hard to explain very quickly, but you can see this biphasic effect where the organism is taking over here on the right side on the day after the remedy is given. That's what we rely on in homeopathy. We throw a remedy in as a signal, and then we let the body take over from there. So in summary, if the right remedy, you are sending a salient signal to that organism. The organism, if diseased, responds by trying to adapt to that signal. and in the course of doing that, amplifies the responses, and this leads to healing and a healthier organism. So I will stop there with that particular point. It's just a summary here. We have a very complex set of materials that are source and silica coated. They have signal properties. They have physiological signaling effects on the nonlinearity of the system. And what is modulated is the functioning of the stress response biology that leads to the healing response. So I will stop and let folks take over with questions.
Well, thank you very much, Dr. Bell and Dr. Fisher, for a very enlightening, complex, and very challenging talk. I must admit that I'm one of the 2%. I carry around a spray bottle of allergy remedy and use that to address the time when I'm sniffling and coughing and things that just come up. So I'm a user, and I'm looking really at the practical side and also understanding what Dr. Bell had just said in the last point is that we are dealing with a very complex system. And frankly, I really don't, after even 40 years in practice, I really do not fully understand the healing process, what we go through when we take a drug. And what is that doing? Is that really the drug, the biochemical intervention, or this placebo effect and this, practitioner effect in the whole process? So to me, it's a very, very complex issue. I do have a question on the practice side for Dr. Fisher that I did not see what's the percentage of people in the UK that uses homeopathy, and because it's covered by your national health. And does that make a difference between what's being used in UK versus in the world?
Can you hear me?
Yes.
Oh, good. Well, yes, it is available on the National Health Service, although that is currently being challenged. But that's a small amount of the total provision. And there was a thing on the. I did show the numbers, which are the estimates vary quite widely from about 3.5% to 9% in the UK. There's two quality studies. One says 3.5%, one says 9%.
So that is still very small. So I'm just wondering, what is the sort of the blockage for more people to use it? And obviously, given the cost effectiveness that you said, it seems like more people should be using it.
Yes, well, I mean, there is an attack on homeopathy. There is a global attack on homeopathy, driven by skeptics who refuse to believe what Iris has just demonstrated to us, that these very high dilutions, could possibly have effects. So that is, and the history of homeopathy in the United States is quite an unhappy one, as you may know. If you go back to the 1890s, maybe 20% of all US physicians were homeopaths. And that was, that number dropped drastically after the Flexner report of 1910. It's now recovering. Yes, I think also there is a certain element of, we said with friends like that we don't need people. With friends like that we don't need enemies. That certain causes get attached to homeopathy, anti-vaccination particularly. Homeopathy is not anti-vaccination. Interestingly, homeopathy was invented in the same year as Jenner first practiced vaccination, 1796. So Hahnemann, the founder of homeopathy, lived through the introduction of vaccination and thought it was a wonderful thing, was 110% in favor of vaccination. He made a famous statement about how before vaccination, a city would lose half to 3/4 of its children to this wretched pestilence, meaning smallpox. So it's a complex thing, and as a cultural element, it's quite more popular in France, Germany, very popular in India, but growing in places where it doesn't have roots, South Africa or Japan or Iran. All those places, homeopathy is taking off.
So Janet, do you have questions from the audience?
Yes, we have one question. Do homeopathic medicines have a number needed to treat calculation like pharmaceuticals?
No, the short answer to that is no. At this stage, there simply isn't enough data. Well, actually, that's not quite true. Actually, there is one study that involved a number needed to treat calculation, and this was a study with the endpoint was a very hard one. The endpoint was alive or dead. This was a study done in the University of Vienna in Austria, study of use of homeopathy in patients with life-threatening sepsis. And there was the endpoint was alive or dead at 6 months. And the number, so it was 75% alive with homeopathic treatment, 50% alive with plain standard treatment, so add-on homeopathy. So a number needed to treat of 4. And the endpoint, the thing that was being treated, was dying or not from sepsis. That is the only one I can remember offhand. But it's a good quality study. The first author is a guy called Frass, F-R-A-S-S, done, as I said, in the University of Vienna, Austria.
So, Dr. Bell, to me, the science of medicine resides really in the biochemistry of medicine. So everything is really based on the biochemical model. So it is very hard for conventional scientists and doctors, physicians, to really understand the sophistication of the homeopathic model, and with everything residing on the nano environment. So I know, I just don't know how does one come that.
Well, it's an excellent question. I believe that in the research world, we start opening up our minds as we run into concepts. So, number one, I think in conventional medicine, we're increasingly recognizing, at least in cancer care, that we have to do personalized medicine, partly based on the genomics of the individual with the cancer, until we can identify the best agents to treat that person with. So I think that concept is beginning to take over in conventional medicine. For the other, there is a growing literature stemming from the hormesis research world, which was actually historically very frightened of being involved with homeopathy at all, because people wrote off the concept of hormesis because it was somehow associated. Low doses can't do anything. And so it's associated with homeopathy, therefore it's bogus as a concept. Hormesis is being studied more and more. There are thousands of published papers now demonstrating that it occurs. And many of those studies are using plant materials from foods that we eat. So there are many studies, growing studies, on plants that actually can produce hormesis in the bulk form. So we're talking about basically low doses in the bulk form of the plant material. And if you take it that next step, if people start looking, because they are now making some of those plants into nanoscale engineered nanoparticles, you're going to start seeing people showing hormesis with nanoparticle forms of things like resveratrol and other materials of that type. If you look at on the bibliography, I hope I have it here. Dr. Calabrese published a paper on mechanisms of hormesis, and he specified some very particular cell signaling pathways and cell receptors that are under study in hormesis. My suggestion is that is a starting point for some of the basic biological research.
Janet, do we have any other questions from the audience? Janet.
Hi, that was it.
That was the only, okay. Well, that really, then we can bring this webinar into a conclusion. Before I do that, I want to tell the participants that sign on, there's a 1.5 AMA category 1 credit that you can claim for this hour and a half of webinar. You can claim that through CEPH@gwu.edu. If you have any question, you can always Google the Office of Integrative Medicine and Health, and we can give you the information. I really want to thank Drs. Bell and Dr. Fisher for giving this really a very intellectually challenging talk. And I thank you for all the people that sign on into the cyberspace for this talk. Thank you very much, and hope we'll meet again.